Hello everyone, my name is Kaylee McEvely. Today I'll be talking to you about my microbe of choice, which is Pseudomonas syringae, or P. syringae for short. I chose this microbe because of a couple unique proteins it creates, and I'll tell you about those later. But it's also commonly found in nursery and landscape plants, which I've always found plants interesting, so I decided to do a little research on this microbe. So, P. syringae is a common plant pathogen in the US, so you've probably seen it on your plants. As I said, it's common in nursery and landscape plants, such as tomatoes. It invades damaged plant tissue and produces toxins and proteins, and the toxins that it produces then kill the surrounding plant cells of the host, where the bacteria can multiply, and unique proteins create ice crystals, which severely damage the plant cell, but they're pretty cool. So P. syringae, a little more information on it. Sorry, the lighting's bad. But as I said, it's a common extracellular plant pathogen. It's commonly found in tomatoes. Here's a close-up image of P. syringae. As you can see, it's a rod-shaped bacteria. It's pretty close, you can't really tell, but this is a picture of the bacteria invading ovaries of a tomato. And it is carried through the air, rain, insects, infected plants, and stationary object carrying the bacteria. You'll notice I didn't say it's carried in soils, which was the original thought process of a lot of studies, but it was found out that P. syringae actually only infects the phyllosphere of a plant, so that's above the ground. So it can't really be contracted through the soil. And P. syringae interacts with the host plant cell by injecting various affecting proteins, and it's pretty cool. It can actually specify based on the host, so it differentiates on a tomato versus a woody plant. So here's an image of the ice structures that P. syringae creates. One of the studies I found interesting on this was conducted by David Sands, who recognized P. syringae acting as an ice nuclei, using specific proteins on the surfaces of host plants by assisting water molecules to turn into ice. So that's really specific and unique about this bacteria. And it's actually used to assist in making artificial snow that we've all probably played in. So this bacteria can slow down the melting process and allows us to enjoy snow for a longer time and in a warmer climate where snow might not always stay around. This trait also makes P. syringae very important for water recycling in the ecosystems where it is found, because it can control these ice structures of the plant. And then a little bit of information on the metabolism of P. syringae. Though other Pseudomonas strains, metabolic processes do not require oxygen, they can be anaerobic or facultative. P. syringae are obligate aerobes, which works out because they're on the outsides of plants. They can specialize their metabolic processes for each plant species. So, as I said earlier, it differentiates depending on the plant. So that's pretty cool. They have a bunch of various strains and various metabolic processes that they undergo. Though they are able to produce a number of secondary metabolites and use multiple compounds absorbed from the nutrients of their host as main energy sources for metabolic processes. The reduced nitrate can be used as terminal electron acceptors to maintain oxidation and to enhance growth for the bacteria. And that makes it an important contributor to the nitrogen cycle as well. As I mentioned earlier, it's also very important for the water cycle of the environment. And for that, it needs everything that you would usually need for nitrogen reduction when creating ammonium, and iron, ATP, and then what's also unique about it is that it undergoes negative oxidase reactions because it lacks a specific cytochrome C oxidase to oxidize the reagents in the respiratory electron transport chain. And then here's another image of the bacteria invading a tomato species. As you can see, the dark spots is where the P. syringae is coming in, and eventually will take over the host if it's successful, absorbing all the nutrients of the host and eventually killing it. Further understanding P. syringae can help understand what makes bacteria pathogenic to its host and how these bacteria have evolved and conformed to their specific hosts can be analyzed through the differences in comparing their metabolisms. There's extremely evolutionary and very elastic. They can change a lot with their hosts. So, yeah, it'll be pronounced. Oh god, I need to edit that out. Okay. And that is all. Thank you so much for listening to me talk about my new favorite microbe, P. syringae. I hope you enjoyed it and learned a lot about their metabolisms and how they contribute to their environment and how they can conform to their hosts. Are there any questions? You? Yeah, you?
Hi. I had a question. Is this microorganism pathogenic to humans?
That's a great question. Wow, that's a great question, young kid.
Participant.
It does not exhibit any pathogenic characteristics towards animals or humans.
Oh wow, thanks.
Great question though. Well, if there's no more questions, that's all I have for you today. Thanks a lot for listening. Remember to like and subscribe if you enjoyed the video. And if you do have any questions, just comment them below. Thanks for watching, guys.